I'm Gordon McVie from ecancer, and I'm reporting on the plenary session of a very exciting meeting in Barcelona. This is the 26th meeting of the EORTC, the National Cancer Institute of the United States, and the AACR, that's the American Association for Cancer Research. I'm feeling my age because I was one of the founders of the first of these meetings in the late 1980s. I was speaking on Eureka and launching the Eureka project. It's an FP7 project on reusing clinical data and getting hospital data that we have everywhere more accessible for asking new clinical research questions. I went through the problems of setting up this sort of really critical project funded by the European Commission. Things like data sharing, moving data from one hospital to another, massively difficult in some countries, and the legal issues get in the way of the science too often. There's security issues, obviously, and a lot of hospitals distrust data warehouses. There are very few, but there are one or two issues surrounding intellectual property. The big issue is data protection, and for Europe, of course, half of the 2,000 people at this meeting in Barcelona are not from Europe, so this was not important for them, but for the Europeans it certainly is important, because the European Parliament have passed an act which will kill data sharing and retrospective studies with tissue from biobanks. And future studies which are not specifically laid out in an informed consent form which the patient signs. I urge you, if you're listening to this in Europe, to lobby your MEP and your MPs to get this changed. There's still time to change the legislation. It really could be devastating and could be the end of using big data in Europe, and this would be a tragedy. The plenary session was extraordinarily good. It was chaired by the chairman of the EORTC, Roger Stoop from Zurich, co-chaired with Josep Tabernero from Valdebron here in Barcelona, so I suppose he was the host. The early clinical trials for testing new agents, of which we have a superfluity at the moment, were reviewed by Dr. Bedard from Canada, and he honed in on several really interesting, really exciting novel techniques which are started off being called basket techniques. The first one was done by Beppe Giacconi at the NCI, and this was 5 different drugs from 5 different companies put into one trial, and the determination of which of these arms, which of these drugs you got, was based on the molecular diagnostics, and that was really exciting. But since then there have been a number of novel designs such as this. We're aware of all these drugs which are waiting to be tested. We're aware of the need. We know that patients are in a hurry. They want the latest things now. They hear about personalized medicine, they hear about genome, and they hear about targeted drugs, but they're not getting them fast enough. So that was very important. And then addressing the issue of speed, Fabrice André from Gustave Roussy in Paris reviewed the ways that we could move from testing one gene and one target and one drug to looking at sequences of genes and looking for other ways of arranging the data which we get out of the molecular diagnostic lab. That was most interesting, and of course Gustave Roussy has probably one of the leading institutes in Europe to take this approach forward from the molecular diagnostic lab to the new drug testing. After that, there was a remarkably inspiring talk from Dr. Dienstmann from SAGE of the United States, and he challenged the textbook, the idea of one gene, one drug, and he said that as far as he was concerned, looking at supervised clusters of genes in prospective or retrospective data was not bringing up sufficiently robust models for taking drugs into trial and being more sure that we're getting the right approach for patients. And he was looking at unsupervised clustering, and he ran through a number of really interesting ways which Eureka could use of looking at of the genomic data and letting the data speak for itself rather than prejudging what might come out of it. And then there was a very interesting talk from Dr. Mardis from the MD Anderson in Texas, and she reviewed the genomic discovery process in acute myeloid leukemia, remarkably short of gene targets, interestingly enough. And she raised a few questions about whether liquid biopsies, which are very trendy at the moment, would indeed offer that much more of an advantage, because it hadn't done so well in AML. And then the last talk was from EORTC itself, from the director-elect, Denis Lacombe, and this was a first-class talk on SPECTA. SPECTA is, as far as I'm concerned, the most exciting initiative that EORTC has embarked on. It's 50 years, certainly in the 25 years since I was president. And it's a wonderful collaboration with the Sanger Centre in Cambridge, looking for patterns and looking for basing trials faster, baskets of trials, and a variety of really interesting innovations. Solid security, good data warehousing, all the best statistical techniques. And it was really a first-class session altogether. So if you can't make it this year, then I recommend that you come to Munich in 2 years' time for a repeat run of this excellent meeting.